Good morning students, I Ram, myself Sridevi Rao will be continuing the lesson. We learnt about, yesterday in the previous video, we learnt about mitosis. There are two divisions we learnt about mitosis. Mitosis, where does it occur? It occurs in somatic cell and stem cells. I told you in the previous video itself. Somatic cells and stem cells means they are the all other cells which are other than the reproductive cells are called as somatic and stem cells. And meiosis occur in germ cells. Germ cells means they are the reproductive cells. And somatic cells are divided by and stem cells divide by mitosis. We learnt about mitosis. Mitosis is completed through main two steps. What are the two steps? We learnt about karyokinesis. What is karyokinesis? There are four stages of karyokinesis, and we are going to learn about cytokinesis. Karyokinesis means it is nuclear division. Okay, and cytokinesis means cytoplasmic division. Under karyokinesis, we learnt about prophase, metaphase, anaphase, and telophase. What happens in prophase? We learned that the nuclear membrane slowly starts disappearing. The chromosomes will become short and thicker. Okay, and then the centrioles will duplicate and they will move to the opposite poles. Okay, and the chromosomes will start pairing with the sister chromatins. Sister chromatins means same type of chromosome. And before the before any cell division, the chromosome number will become double. Okay, now when it is in the prophase, after prophase comes the metaphase. In the metaphase, we learn that the chromosomes have become thick and short, and they are clearly visible, and they are arranged in the equatorial plane. It is in the central part of the cell, and there are spindle fibers which are attached to the centrioles and the centromere of the chromosomes. Okay, and in anaphase, the centromere divides into two parts, and half of the chromosomes go sister chromatins, that goes up and half comes down, and they look like bunch of bananas. And in telophase, there is division. There are two nuclear membrane. There are two cells, daughter chromosomes present, and it will be similar to the parent because there is no any crossing over taking place, and two diploid cells are formed in mitosis. Understood mitosis? Now, after this comes the cytokinesis. What is meant by from the name itself you can make out it is cytoplasmic division. Okay, the cytoplasm divides by cytokinesis, and two cells are formed which are called as daughter cells. Okay, after cytoplasm divides, there are two daughter cells formed separately. In this process, how it happens is a notch is formed in the equatorial plane of the two cells. See, in telophase, it was attached. Okay, two cells they were formed where they were attached. In this process, a notch is formed which in the equatorial plane of the cell, which deepens gradually, and thereby two cells are formed. So in the middle, what happens? There is a notch formed. You can see here in the diagram. This was the above one was telophase. So slowly it becomes elongated, and a notch is formed in the middle, and there are two separate daughter cells which are similar to the parent that comes out. Okay. In the case of this is in case of animal. In the case of plants, what happens? Instead of a notch, a cell plate is formed exactly along the midline of the cell, and the cytokinesis is completed. So in the case of plant cell, instead of a notch, a cell plate is formed in the middle. Okay, in the midline of the cell, and like that two cells separately they are formed in in mitosis. So I think you are thorough with the mitosis. Now coming to the next division is meiosis. Okay, why mitosis is, I forgot to tell this point, why mitosis is needed for the body. It is necessary for, first itself we discuss, for the restoration of emaciated body, wound healing. Any wound in your if there is any cut, you can see next day it slowly starts covering. How it starts covering? There is multiplication of cells. For this, mitosis is needed, and formation of blood cells also. For all this, there is multiplication of cells, so there is no any fusion of gametes or there is no fusion of any chromosomes in this just as a multiplication of cells. And this one, so for all this, for restoration of emaciated body, for wound healing, as well as for formation of blood cells, mitosis is essential. And then for the growth of the body, we saw all this in the previous video. Now coming to the next part, meiosis. Meiosis is completed in two stages. What are they? One is meiosis 1 and meiosis 2. See in your textbook, detailed description of meiosis 1 and 2 is not there. How much is there in your textbook? That much if you learn, that is enough, because you will be learning meiosis 1 and meiosis 2 in detail when you go to standard 11. Okay. Now we will learn in short what is meiosis 1 and meiosis 2. First we will start with meiosis 1. See, in meiosis 1, there is recombination or crossing over between homologous chromosomes. What are homologous chromosomes? Chromosomes, that means chromosome from one is from the male and one is from the female. That is paternal and maternal. One chromosome in mitosis, it was the sister chromatid. That is the same type. But in meiosis, there is homologous chromosome. One is from the male parent, another one is from the female parent. And thereafter, those homologous chromosomes are divided into two groups, and thus two haploid cells are formed. In this meiosis, there is a recombination or crossing over. I'll show you what is meant by recombination. See here. See this, in meiosis there are same way 5 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. 4 phases are there. Under prophase there are 5 steps. Okay, under prophase 1 there are 5 steps: liptotene, zygotene, pachytene, diplotin and diakinesis. These names are not given in your textbook, but this is for your extra information. Under prophase 1 of meiosis, there are 5 stages. For 5 steps, they are liptotene, zygotene, pachytene, diplotin and diakinesis. In this prophase 1 itself, you can see color of the chromosome. One is blue, another one is red. That means one is of male and one is of female. So what will happen is they will come close by each other, and in the last stage, in diakinesis, you can see there is crossing. After this process, there is crossing over. That means half of the blue will be connected to this red, and red will go to blue, blue will go to red. That means there is crossing over of the chromosomes taking place in this prophase 1 of meiosis. Okay, this is the picture in your textbook. Okay, 1, 2, 3, 4, 5 they have named there. You can see two are blue in this and 2 are red in this in the first. Then they pair with each other. Okay, one blue, one red, one blue, one, like they pair, and they become thicker and shorter. And the fifth stage, there is after that there is crossing over of the both the colors, that is paternal and maternal chromosome. There is crossing over, and you can see this in the fifth stage. After this, the metaphase, same way spindle fibers will be formed. All these chromosomes will be arranged in the equatorial plane. In the metaphase, the same thing happens. See, you can see in the anaphase itself, slowly it starts dividing. The spindle fibers pull the whole chromosome. That means the centromere does not divide over here. The one set of chromosomes fully come down and one set of chromosomes goes up. Okay, that means only half the number of chromosomes are present in telophase. That means haploid number. You get 2 daughter cells which are haploid. Haploid means only half the number of chromosomes are present in each of the daughter cell. So after meiosis 1, you get 2 haploid daughter cells. In mitosis, it was diploid because the number of chromosomes does not change. Because it is divided into half and half, they go up and down. Here, the full chromosome, the centromere is not divided. So, the one set of chromosomes goes up and one set of chromosomes comes down. So, there are 2 daughter cells which are haploid. You can see here. Okay. So, in the end of meiosis 1, you get 2 haploid daughter cells, chromosomes. You get 2 cells. Then these 2 haploid chromosomes, which are daughter chromosomes, take part in meiosis 2. So, okay, meiosis 2 is just like meiosis 1. Okay, it is just like meiosis 1. That means, so I'll repeat what happens in meiosis 1. In meiosis 1, and there are 5 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Under prophase 1, you can see there are 4 stages. What are the prophase 1? There are 5 steps in prophase 1. One is liptotene, zygotene, pachytene, diplotene, diakinase. Only the names of it you can learn. What is that? Liptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, so in this what happens, you can learn that is only crossing over of, you can see, of the homologous chromosomes. Crossing over between homologous chromosomes takes place, and the homologous chromosomes are divided into 2 groups, and 2 haploid cells are formed. In this node diploid, haploid, that means half the number of chromosomes are only present. Okay, so this was the prophase 1 stage. It is what 5, it is divided into 5 steps: liptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, in this crossing over, the blue color will slowly come to red and red to blue. So there is crossing over of the homologous chromosome. Homologous chromosomes means one from the male parent and one from the female. That is not the identical one, it is the opposite ones. Okay, so this is in your figure in your textbook. 1, 2, 3, 4 prophase 1, and then metaphase 1. It is similar. You can see they are arranged in the equatorial plane. Then comes the anaphase. That means the full chromosomes, half will go up and half will go down. You can see how their combination has taken place. In the blue, red color is there. In the red, blue is there. All are different from each other. It will not mix up the similar way. So there are 2 haploid daughter chromosomes present in telophase 1. Understood meiosis 1? Okay, now we will go to meiosis 2. Meiosis 2, it is similar to meiosis 1. No, mitosis. It is meiosis 2 is just like mitosis, same way. But 2 daughter chromosomes will take part in this meiosis 2. Same way, you can see 2 daughter haploid new daughter cells formed in meiosis 1 undergo division by separation of recombined sister chromatid, and 4 haploid daughter cells are formed. See in this, how many haploid cells take part? 2 haploid cells, daughter cells, which are formed in meiosis 1 take part in this meiosis 2. And same way, this 2 daughter cells will go as we have seen in mitosis prophase. That is prophase 2, metaphase 2, anaphase 2, and telophase 2. And after that, you can see the same similar way. You can see prophase 2, the 2 daughter chromosomes, they become thicker and shorter. Okay, they are in pairs now. The haploid, they will become thicker and short, and their nuclear membrane will disappear. The centrioles will be opposite poles, same way, but in 2 cells. Then again, in metaphase also, in 2 cells also, they are arranged in the, you can see then, they are arranged in the equatorial plane. In the middle, the spindle fibers are formed, but the chromosomes are not sister chromatids. It is the combination, recombined homologous chromosomes. That is a different pairs. There is already recombination taken place. So it is arranged in the equatorial plane. So it is similar to mitosis. In mitosis, we saw the chromosomes become thicker and shorter, and the nuclear membrane disappears, the nucleolus disappears, the centrioles which are present, they'll duplicate and they will move to the opposite poles in prophase. So same way in prophase 2 in meiosis also, the same thing happens, and you will get like this, thicker and shorter, which are clearly visible. Then when they go to metaphase 2 also, they are arranged in the equatorial plane, and their spindle fibers are formed. Okay, so the same thing as metaphase of mitosis. Then the next stage comes the anaphase. This is anaphase. You can see They are the spin. They are half of the chromatids. Again, the centromere divides, and you can see half. You can see only a V part. See, the half of the chromosome that goes up and half comes here, like the mitosis of anaphase. So there are, you can see here, half of the chromosome which was like a, in the previous figure you saw, it was like this. It was like a full chromosome, and when you come to the anaphase, you can see half of it is pulled up and half of it is pulled down, and you can see on the telophase there are 4 haploid daughter cells. At the end of telophase, how many haploid? You can see there are 4 haploid daughter cells formed in this telophase. Okay, so what all we learned, children? We saw, so in this type of cell division, you can see how many daughter cells are formed. Haploid means only one set of chromosomes. Diploid means 2 sets are present. So in this type of cell division, 4 haploid daughter cells are formed from one diploid cell. Okay, now during the cell division, crossing over occurs between homologous chromosome, and thereby genetic recombination occurs. That's why there are differences between parents and offspring. That is genetic recombination. That is crossing over of parents. Okay, parents chromosome, one maternal and paternal, one from mother and one from father. That's why there is genetic recombination. That's why there are differences between parent and offspring, or else all will look identical. When you take mitosis, the daughter cells will be similar, identical to the parent. But in meiosis, because of the crossing over of the homologous chromosomes, and that's why you have genetic recombination, and that's why we have differences between parents and offspring. Even your siblings, your sister and brother will not be identical to you, because the crossing over differs in each chromosome. Okay, that's why. And so the crossing over that's why there is genetic recombination occurs. So hope you all understood, children. Let us revise now. So due to this, all 4 chromosomes are genetically different from parent and from each other too. So that's why when we get, all the 4 will be different. When you see the figure of that also, you can make out. Okay, I'll show you the figure of the telophase. You can see the 4 haploid, all the color combination is different in telophase. You can see there are 4 haploid chromosomes present, but for haploid cells, but all the 4 are different in their colors itself. See, some of the combination is different from or most of them are different. That's why we get all the 4 different haploid nucleus. You can see. See, due to this, all the 4 daughter cells are genetically different from parent cells, and thus from each other. So all are not identical to each other, are not to the parent also. So for this, where this meiosis is used in gamete production as well as in spore formation. So where it is used for, we saw mitosis producing, for restoring emaciated body, for formation of red blood cells, for the growth of the body, mitosis is needed. But for meiosis is needed only for gamete production, that is for in reproduction process. As in the plants, it is a spore formation. Okay, so I will revise what we had learned. We learnt about meiosis 1. We saw about the 4 in the previous video. We learnt about the karyokinesis undergoes. There are 4 stages of karyokinesis: prophase, in mitosis. First we will revise mitosis, and then we will go to meiosis. In mitosis, under karyokinesis, there are 4 steps: prophase, metaphase, anaphase and telophase. In prophase, you all know, the chromosomes become thick and clear, short, and the nuclear membrane disappears. Nucleolus starts disappearing, and the centrioles move to the opposite poles. They will duplicate and they will move to the opposite poles. And in metaphase of mitosis, what happens? They are arranged, the chromosomes become they are visible, clear, they are very clear, and they can see them arranged in the equatorial plane and the central part of the cell, and the spindle fibers are formed between the centrioles as well as the centromere of each chromosome. Then the spindle in anaphase, what will happen? They will pull apart and half of the chromosome set, that is the centromere divides. Okay, there is division of the chromosome and half part of the chromosome goes up and half the part comes down, and then they look like bunch of bananas. And and in the telophase you can see there are two sets of daughter nucleus formed, but they are not divided from each other, they are attached. So nucleolus is formed, nuclear membrane is formed in four, and there are two diploid daughter cells formed. And after karyokinesis, cytokinesis takes place, that means division of cytoplasm, and then you get two different daughter cells which are similar to parent. But in meiosis, it is into two stages, meiosis 1 and meiosis 2. In under meiosis 1 we saw there are prophase 1, metaphase 1, anaphase 1 and telophase 1. In that, in after telophase 1, how many daughter cells you get? You get two daughter cells which are haploid. That means only half the number of chromosomes are present in the end of telophase 1. These two daughters which are formed in meiosis 1 will take part in meiosis 2, and it is this meiosis 2 has got four stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. In this, what happens? Same way like mitosis itself, but in two cells. Okay, and they are arranged in their chromosomes become thick and short, centrio, both similar like prophase 1. Prophase of mitosis. Then in metaphase, they are arranged in equatorial plane, spindle fibers are formed, etc. And in anaphase, the centromere does not divides into two, as in mitosis. The centromere divides into two, and they divide into up and down, they go up and down, and four daughter haploid cells are formed in the end of meiosis 2, which are not identical to the parent and from each other. So this process is used in reproduction, that is for gamete formation as well as for spore formation. Hope you all understood this lesson, children. We have taken 5 sessions to complete this lesson. Go through the lesson, read the lesson and mark the important points. This lesson is very important. So, hope you all understood. Okay, children. Thank you. Sai Ram.